All right. Good morning and welcome to the March Epilepsy Grand Rounds at GW. Before I introduce our speaker, please remember to claim your CME by texting the number at the end of this talk, and then you'll get CME credit automatically. Our speaker today, I'm delighted to introduce him, Dr. Nitin Tandon. He's a professor of neurosurgery and vice chair of neurological surgery at University of Texas in Houston. He's also chief of the epilepsy surgery program there. Nitin earned his medical degree at the Armed Forces Medical College in Pune, India, and then pursued his clinical training at University of Texas in San Antonio. Before going for an epilepsy surgery fellowship at the Cleveland Clinic. He then returned to Texas to Houston, where he's on faculty at the McGovern Medical School since 2003. He has done extensive research and has published his findings in some of the most esteemed medical journals, including Nature Neuroscience, JAMA Neurology, as of yesterday, I heard. Obviously, Journal of Neuroscience, Human Brain Mapping, Cerebral Cortex, among numerous others. His research focuses on intracranial recordings and brain mapping. He tackles higher cognitive functions through different methodologies, including intracranial recording, brain mapping, and also functional neuroimaging. He has secured extramural funding from federal sources, National Science Foundation and NIH for his research, and recently has a multi-center grant to study reading in the human brain. If you ask why would a neurosurgeon be fascinated by language and do all this exquisite research to understand the neural mechanisms of reading and other language processes, I'm delighted to tell you a secret today that Dr. Tandon is also a poet. Please join me in welcoming Dr. Tandon.
Thank you so much. Thanks for having me. Well, good morning, and thank you for joining. Thank you for that kind introduction, Mohammed. As you all know, there are links to this lecture, and there are also these upcoming events. I don't have any disclosures relevant to this presentation, and at the end of this presentation, I hope you will be able to benefit from these learning objectives. This is the process for claiming CME credit, and this will show up again at the end. So as Mohammed said, so why would a neurosurgeon be interested so much in language? Well, firstly, it is very relevant to what we do as clinicians. As you all know, there are many cases of epilepsy and brain tumors where there's a interplay between the organization of language and where the lesion or the abnormality that we want to target is. But that is the underpinning, that is the core purpose. But the other purpose, just as importantly, is from a purely scientific aspect to understand this process, which is very uniquely human. We think language originated about 250,000 years ago. There was this big emphasis that it coincided with the emergence of the FOXP2 gene, but that has been thrown into some doubt recently. And another reason for us to study language in humans is language is not really assessed in non-human species. It's not to say that other animals do not have any kind of language, but they certainly don't have the richness of language as we do, and it's much harder for us to study. But more relevantly, language disorders or aphasia are a huge problem, and the estimated incidence is about 80,000 new individuals a year, and a prevalence of around 1 million people. The prevalence of aphasia, therefore, is greater than that of Parkinson's disease or of muscular dystrophy, disorders that we spend much more time on studying. And one of the reasons we don't study aphasia is because we can't do anything about it. So if someone has a brain lesion where they are rendered mute or they are rendered unable to communicate basic needs, we shrug our shoulders, point them towards the rehab doctors, and walk away. And we hope that this kind of work, the basic understanding of the wiring of language systems, provides us someday soon with the underpinnings of creating technologies to create prosthetic devices for these humans. And I would also say that many of us, I think even at dinner yesterday, we have this problem of not being able to remember names of people. we struggle in contexts that are unfamiliar to us. The same individual, if I ran into Muhammad someday at the Guggenheim, I might not recognize him instantaneously. It might take just a slight bit longer, and if it was somebody else in the room that I've only met once or twice, I almost certainly wouldn't remember your name. But in this context, I would. And so there are a variety of reasons why we have this problem of finding words. And this problem of finding words is a sort of almost a normal human process that gets more exacerbated as we get older. Language mapping has been performed by various people for well over a century. This is Paul Broca, Carl Wernicke, and in fact, even before Broca, there was a gentleman named Mark Dax who described Broca's aphasia. These are the brains that Broca studied. And then, of course, here is Foster and Penfield. Foster was a German neurologist to a neurosurgeon. Here are the maps produced by Ogerman. This is C.J. Ogawa, who created fMRI, the first big scanners at Minnesota, and Mark Rakel and Peter Fox, my own mentor in functional imaging. And this evolution of technology from lesion to imaging has given us very rich maps of the organization of language and has led to the development of various language models from these techniques. But these models have very little integration between them, and very often their derivation is driven by behavior and not by actual data flow between brain areas. And therefore, such models that show us that there are brain areas that are active in a given process don't necessarily answer how and why the brain works a certain way. They can tell us, of course, what happens. They can tell us where things happen, but very little also about what and when actually happens. And it has led to the birth of these kind of models that we've all seen, the Lichtheim model, which is several decades old, almost 80 or 90 years old at this point, which is just like this, very serial, very linear, very modular. And I think most of us understand the brain doesn't actually operate this way, that one area doesn't just have feed forward or feedback interaction with something else. And instead, we feel that word production probably functions in this way, that if there is a particular word that we are seeking to use, its phonemes and its features are bound together instantaneously for us to then create that particular word. But again, this is another model that is born out of psycholinguistic behavioral determinations rather than actual data. So what we need is data really at this scale, at the nanometer scale and the millisecond time resolution. And the closest that we can get to this today is with recordings in the human brain, somewhere around here. And of course, you could argue that you could do microwires and micro-ECOG, but that would be studying a very small number of cells. And pragmatically, at this millimeter resolution and at the millisecond time scale, there's a relative sweet spot that we can use to assess what is happening in the brain. So what ECOG does is provide us data from awake behaving humans with high temporal and spatial resolution, coverage of many areas potentially involved in language, allows us to critically assess how these processes in the brain work. And perhaps, just perhaps, I hope, by the end of at least my career or in the next decade or so, we come up with a new way of describing brain processes. A new terminology, a new dictionary is created to describe what actually happens in the brain, rather than simply using and imposing terminologies that we have from psychology or from other branches of science. So stepping back a little bit, we're talking about intracranial recordings. And intracranial recordings in humans are not exactly straightforward. I reviewed this paper that itself is a systematic review and a very thoughtful meta-analysis of 61 papers with 5,600 patients that met the criteria for this particular review. The authors divided the patients in this review into two groups, two cohorts, before 1995 and after 1996. Nothing magical happened in 95, but That is around the time when imaging, frameless stereotactic navigation systems, formal epilepsy surgery training programs, and the use of operating microscope and operating techniques were much more standardized. And before that, perhaps there was much more variability. And I want to draw your attention to various numbers here. As you can see, there's a huge difference in the risk of transient deficits. So before 1995, the risk of transient deficits with the temporal lobe operation was 42%. And after 96, it became 5%. Of course, the risk of permanent deficits is very small, less than 1%, and I think that is borne out by all of our experience even today. These risks are much greater for extratemporal and multilobar operations. But the one risk that doesn't go down with time, but actually goes up, is the electrode placement. And it was 2%, risk of infection went up to 4%. The risk of bleeding in the head went from 2% to also 4%. So I would submit that we have now an approximately, or had, an 8% risk of complications from what is essentially a diagnostic procedure that I depend upon to use to collect scientific data while patients are waiting to have seizures, but still a procedure that has some morbidity. And this pushed us and pushed others to think about different alternatives. I would also submit that the reason why this number has increased is for two reasons, perhaps. The patient type that was assessed by various groups evolved, where we were studying more complex patients, patients who'd had prior surgeries, patients who'd had, say, prior brain infections, scars in the cortex, risks of various kinds, more extensive or more invasive electrode implantations. But also that we transitioned from placing mostly depth electrodes in the early 80s and mid-80s to almost entirely grids in the late 80s, 90s, and 2000s. So enter Jean Taylorach. He needs no introduction to this group or to most epilepsy circles anymore. He was originally a psychiatrist, as some of you might know. He was convinced, of course, that there were organic relationships between psychological disorders and functions of the human brain, and famously said that psychosurgery is far too important to be left alone to the neurosurgeons, decided to become a neurosurgeon, trained as one, and then developed this novel concept of three-dimensional network-based evaluation of epilepsy. And worked then with Pierre Tournou to develop an anatomical atlas derived from a 60-year-old French woman post-mortem, in which he labeled Brodmann areas and characterized this as the standard space in which he would implant electrodes. So in his lifetime, transition from psychiatry to neurosurgery, created the Taylorach space, a standardized space for reference of the human brain, and SEEG. And this paper, the earliest paper on SEEG, is published with several patients in 1965, data collected in the 50s and early 60s, very orthogonally placed electrodes guided by arteriography, somehow a technique that did not catch on across the Atlantic. And for a variety of reasons. It was time intensive, it was very driven by semiology. And why? Well, because think about 1960, there weren't even CAT scans, forget about MRI scans. So you couldn't see inside the skull. And you had to gauge where to place these electrodes based solely on the patient's behavior during a seizure, those first few seconds. And this is why the French became such great semiologists. In North America, it was, well, we're going to put electrodes on, and then which side of the hemisphere, or which hemisphere the onsets are greatest in, whether it's anterior or posterior, that's where the grid's going, more or less. And so this was a much more sophisticated way of thinking about epilepsy, I believe. And only recently, somewhere around 8 years ago, this actually traveled across to the US. It was implemented in North America at the Montreal Neurological Institute, of course the French connection, by André Olivier, who has been doing this actually longer than anybody else in North America. But I think a lot of credit also goes to Hans Lurers and Patrick Cheval and Jorge Martinez-Gonzalez for pushing this and bringing SEEG more or less forcing it down the throats of everybody else initially in North America. But I think it has evolved, and I think even the way I do SEEG is different than the way the French do SEEG. We're certainly not constrained to orthogonal placements. We're open to the idea of three-dimensional type of implants in certain cases to narrow down exactly what needs to go or what needs to stay. And it's very azimuth-based placements instead of orthogonal, which means really perpendicular to the skull at any angle, at any entry point, I'm sorry. And then also some groups, including us occasionally, will do staged implants where you have a pretty good sense of which network is engaged in the epilepsy, but then you add more electrodes as you understand it better. So I think this is quite a bit different from the way SEEG was originally practiced. Also, the electrodes have evolved, and we all almost now use 0.8 millimeter diameter electrodes, which are much thinner and have half the cross-sectional area of a 1.2 millimeter depth electrode. This is just a summary of the accuracy of implantation using a robot, which shows that the mean error is pretty small, even when very angled trajectories. So when you see angled trajectories of 50 to 75 degree angles to the skull, that's a very steep angle to the skull, you can still get very close to where you intended to get. So this paper, as Mohammed alluded to, just was out of embargo yesterday. Just published in JAMA Neurology, where we compared our own experience with grids versus SEEGs. So this is 260 patients that I've operated on over somewhere around 12 and a half years, 13 years. We stopped data collection and analysis about a year prior to this point. And as you can see, this is the overall trend. We were doing a certain number of grids. We started doing SEEG in blue here, and SEEG numbers went up and the grid numbers plateaued. We still do some grids. This is cumulative numbers, of course. We still do grids, and I'll explain at the end where I think grids still have a role. But mostly we do SEEGs now. And 2017 was half the numbers because that's the midpoint of the year was when we ended data collection for this particular study. On the right, you can see how long it takes us or takes me to do this operation and the learning curve associated with it. So this is for grids, and then this is for SEEG. For the very earliest grid patients, we didn't have data, so there's some missing data points there because it was paper charts back then. It was hard to figure out exactly. But really, you can see in the main, there's a very slight downward decrement, but it really stays pretty stable at around 500 minutes. This is total OR time, by the way. And then on the bottom is the SEEG implantation. So here's the input variables, if you will. How are these? What is different about these two cohorts? So we said the implant OR time 429 versus 322. That's total OR time. The actual implant time, though, is even more dramatically different because these patients are going and getting a CAT scan with contrast, etc. So it's really about 2 hours skin to skin and about 5 hours skin to skin for subdural grids. So it's a huge resource difference there. Of course, we can put more electrodes in with SEEG because they are closely packed. They're also along a single depth trajectory. And 19 patients in the subdural electrode group got transfusions. That's, of course, partially because they have 2 craniotomies within a week or 10-day period, whereas the SEEG patients, only 1 received a transfusion. This is the amount of opiate consumption, the milligrams of morphine equivalents. And I think everybody understands this, that these patients, when you go see them, aren't hurting as badly as the grid patients. But it's almost half, if not quite half, the consumption of opiates. And then major complications, return to the OR for removing a hematoma, an infection that led to prolonged antibiotics or removal of the bone flap, occurred in 6.5% of our patients, somewhat smaller than the published meta-analysis, but not in the same general ballpark. And we've had 0 complications so far. Knock on wood, I'm sure someday that will change with our SEEG population, but it hasn't happened yet. And that's in a total of 121 SEEG operations. Now, it's also important to recognize that this is a diagnostic test, that the placement of these electrodes is done really as a test to determine candidacy for resection. And a good test should tell you who you shouldn't operate on as well. With grids, the threshold to resect is much lower because the electrodes are already in place. You're going to go back, you're going to remove the electrodes, you're going to do something. And often that something is a lobectomy of some kind or some kind of neocortical resection. And so that sort of is baked into the approach of grid electrodes. And so about 91% of patients with grids had resections, and about 73% with SEEG had resections or ablations. Importantly, the numbers undergoing either temporal or neocortical, mesial temporal or neocortical resections were the same, so these groups did not differ in that regard. And here are the outcomes, and I think it's obvious here, but maybe this slide is a bit busy. I'm going to jump to this one instead. So for all the patients who underwent resection or ablation, the class 1 outcomes, and this is a surprise in this data, was that approximately 55% of grid patients had a good outcome, which is, I think, what we would all expect. Grids about 55%. For the SEG, that number was 76%. It was significantly higher, and significant number of patients did much better. This is also shown here on the right as a survivor analysis. And when you look at all patients, not just patients who actually had resections or ablations, but more, if you will, an intent to treat sort of analysis, people who just had the procedure, even so, there was no inferiority of SEEG, even in that context. And in fact, there was an inferiority for bad outcomes with grids, where grids had a slightly greater incidence of class 3 outcomes than SEEG did. So I think clearly there is a evolution in our field. I think most centers in North America have transitioned or are transitioning. Certainly don't. I know that that GW has transitioned to SEEG a long time ago, but this is clearly a way I think the future is going to take us. It is important also to point out in this work that the patients with non-lesional epilepsies were more overrepresented in the SEEG population. So I'll let that sink in for a minute. Even though the SEEG outcomes were better, there were much more non-lesional patients in SEEG than in grids. Of course, we know that we study SEEG, use SEEG to study patients who are non-lesional more often than grid patients. But this just really emphasizes the point that the technique is superior. So why would we do grids in this day and age? I think there are still a few situations. Superficial lesions where you clearly can see the lesion, it's around functional cortex. The patient can't undergo an awake craniotomy, which would be my preferred way of doing this, because an awake craniotomy allows you to not just map, but to monitor, and to monitor the effect of cortical and subcortical resection on function. So if that's not possible because they're anxious or because of whatever reason, then certainly grid implantations are useful. And I think below the age of 3, when the skull is too thin, the youngest I've done is about 4.5-year-old child. But I think there are other groups that are doing younger kids as well. So getting back to where we were, back to what all of this means for our science work. The patients are waiting to have seizures. We have this opportunity while they're in the EMU, if they're willing, to engage them in a variety of different tasks with a laptop computer for a couple hours, 3-4 hours a day, sometimes depending on their willingness. This is what the ECOG data looks like. You've all seen these kinds of traces. You can then decompose this into the 7 canonical bands of ECOG, which is theta, delta, alpha, beta, low, mid, and high gamma. So I broke down gamma into these 3. And as you can see that during a task, there is a decrement in blue here in the low frequencies, but an increase in power in gamma, especially in mid and high gamma. And it is really this difference in between mid and high gamma that looks so small on this graph that is all of the signal that we look at. And as you can see, when you decompose the signal into a spectrogram, which is a representation of its power in different frequencies, on the y-axis is a log plot of frequency, on the x-axis is time. You can see this window of gamma in the dotted bars. And in this case, this box represents a particular time window that we're interested in studying, this increase in gamma power. So we know that gamma holds the key to cognition. There are a variety of papers from many groups that have related various cognitive processes to various functions in the brain, to various spectral changes in the ECOG. I'm sorry. And here's another example of this. So here's an individual with grids over their left hemisphere. I'm going to show you 3 electrodes. There's an early visual electrode, number 1, a electrode in Broca's area, number 2, and an electrode over speech motor cortex in number 3. And as you can see, this is electrode number 1, active very early with visual processing. Electrode number 2, that starts to be active really at about 500 milliseconds and persists just till the onset of articulation, at which point it shuts down. And then here is an electrode over motor cortex that comes on and continues to stay on for a prolonged period of time. So this is just to give you a sense of the differences in brain areas that you can pick up with electrodes implanted. In this case, these are the older style grid electrodes. It's just easier to represent this for explaining where the activations might be. We can then represent this power in gamma and the power in beta on the surface of the brain. So this is the lateral surface and the medial surface of the same individual's brain. And here is where gamma power in motor cortex, Broca's area, is maximal, and there's the visual electrode. As you can see, there's a desynchronization or a loss of power in low frequencies that we can also then represent on the brain surface. And for those of you who are interested in fMRI, this individual also had an fMRI activation. And you can see that it's similar areas, although not exactly. And of course, in fMRI, they're not speaking out loud, so the motor component is missing. But you can see that similar areas in Broca's area, similar areas in the ventral visual stream, in visual cortex are active. So there is a reasonably tight correspondence between the ECOG and the fMRI signal, especially in the gamma band, the middle gamma band mostly. Yet, this kind of ECOG is not optimized to study cognition, and there are problems. The first is coverage. We implant, and we must implant electrodes solely for the benefit of the epilepsy, for understanding where the patient's seizures are coming from. We're not implanting electrodes to study cognitive functions. So there's a coverage problem, because areas that we may be interested in have no electrodes. Also, there is a significant amount of variability across people. These data are scarce. We implant, give or take, around 45 patients, maybe 50 patients a year at our center, some of whom are children, some of whom are people with developmental delays and therefore cannot be studied, and some of whom only have electrodes in for 3 days because they seize very quickly. They go back to the operating room. So it's even at a center like ours, we get several scores of data sets a year, but it's not much more than that. So these are the sort of the clinical constraints. And so this has led to most ECOG papers so far having small series, 5 patients, 6 patients. And I would submit that that's a very poor way of understanding a global human behavior and making certain that we can extrapolate those findings to the rest of humanity. And also there are, in small series like this, there are patient effects. We have, obviously, these brains are not healthy brains, and there are some cognitive limitations, and there may be unknowable modifications of their neural networks that we don't understand. Therefore, the impact of outliers is even greater given the small sample sizes. Also, how do we extrapolate this kind of data to the large data sets that fMRI and MEG and other non-invasive imaging studies have provided us over the last several decades? So for this, we created a platform, software, that allows us to integrate data across people. We call this 3D MIMA, which is a frequentist approach to group analysis that uses the estimate of variability across subjects by converting that to a beta, an effect size, plus a precision estimate for that individual, which is a t statistic, and then combine this in 3-dimensional imaging space. And it provides, this sort of integration provides a result that is even better than a Bayesian analysis of these data, as people have done with fMRI. So there is a MIMA equivalent for functional imaging, and what we have done is purpose that for intracranial recordings. Further, we think it is very, very important to register brains to each other very precisely using surface match technologies or approaches rather than just volumetric transformations. So here, for example, is a single subject, and here is superior temporal gyrus, Broca's area, or part of Broca's area, pars triangularis, and motor cortex. And if you transform this using a volume-based normalization technique into a template MNI brain, for example, you see how some of the voxels make it to the appropriate gyro location and many do not. And then some end up in places where they shouldn't end up. Whereas if you do this using surface-based normalization, which is using the Peel envelope to do the registration, this topology of these brains are exactly preserved. As you can see, this brain and this brain are different in shape, but those regions are the same. Okay? So we applied this technique for all of our grouped analysis, and I'm going to show you many figures. Really this is all a preamble now to what's coming. So before I go further, I talked earlier about SEEG and subdural grids. So this is just to make the point that grids, people feel, gives us this very global, complete coverage. It actually does not. You see the large holes in between very important gyri that are completely unsampled by grids. This is 45 grid patients, and this is 61 SEEG patients. So now the amount of red, which is the heat map for how many patients contribute to a given surface point, are smaller in the SEEG cohort. They are more biased to the temporal lobe, because the temporal lobe is, of course, the thrust of our evaluations in most cases. But it's much more uniform. There's very few parts of the brain that are spared by SEEG. So the answer, I think, is either you integrate large numbers of SEEG patients, or you can even combine grids and SEEGs if you still have some grid data sets. And look on the right, which is 106 patients with everything combined. And for the most part, you have at least 5 patients contributing to each spot on this brain in this average brain space. So to summarize, if you take about 100 patients, okay, it's an ambitious body of work, you should be able to get whole brain coverage, more or less, to study how the entire brain does a given process, which means you'd have to run this experiment, the same experiment, more or less, in that entire cohort. So in our first experiment, we applied this, and we started collecting these data around 12 and a half years ago. These numbers have actually even expanded more since this slide was made a few months ago. As you can see, there's about something like 17,000 total electrodes over the dominant hemisphere, something like 35,000 total electrodes in both hemispheres in 106 patients, with undergoing 30,000 trials of a very simple, this is like the Boston naming test, but the Snodgrass and van der Waart picture set is a little bit more expansive and controls these stimuli for a variety of other features that make some things, like, for example, a banjo is not something that you see every day. So it's a low frequency object. Okay. Similarly, a beetle is relatively low frequency, but it's got high response selectivity. So this could be a bug, an insect, a beetle, etc. So it has various features like this that allow us to probe the space of the stimuli, both in its visual as well as in its semantic attributes, to allow us to determine which parts of the brain are actually doing what. And when we integrate all of this together, here's a map of activation across everybody: visual cortex, Broca's area, and motor cortex. And when you make this into a time movie, a time representation, where now blue and red are simply relative to time zero. Time zero is articulation onset. So this is when they're speaking. Blue is something that happened before speaking, and red is something that happened after speaking. You can see that you have a relatively complete map of language organization. So we're probing this data several different ways. There are a couple of manuscripts in progress. I'm just going to show you some of the power of this kind of analysis. You can then go back to these regions that I showed you were active, and you can pull all the electrodes from all the individuals that contribute to that given region and create joint time traces to see which areas are active when. And later on, I will show you how you can use these data to then understand how the brain is actually operating as a network. In the second type of experiment, we wanted to ask a different question. So you saw a pictorial stimulus here, and you came up with a word for it. But what if I didn't give you a picture, but I still wanted you to come up with something that represents an object? Specifically, we're interested in this conversion of input into meaning and meaning into words. So this lexical semantic interface, you've all seen this in patients with temporal lobectomy. They come back to clinic sometimes and they cannot find the right word. They struggle especially with people's names. In fact, this is almost a part in many cases of the disease itself, where patients with temporal lobe epilepsy tell you that they have a very hard time with people's names because this part of their brain in some way is dysfunctional or diseased or is disrupted by the occurrence of the epilepsy. So I thought it would be important for us to understand where in the temporal lobe this function lives. So for this, we used our image data set like you saw earlier of picture naming. We also used scrambled images, the same image but jumbled up where it was unrecognizable. Naming to definitions, so something like you might hear something a king wears on his head, and you would have to come up with the answer of a crown. And then listening to that same speech but turned around so it was no longer comprehensible. And you ask the question, where is this purple area that takes inputs from either auditory or visual domains? Where does it live that then gives information to motor cortex? So I'm going to jump you through a few slides here. So this is the coverage map. I think you understand this already. These are all the electrodes. This paper is published already in Brain last year, so I won't belabor all of the details. This is the aggregated representation of all of the electrodes. These are the stimuli. And then here is the activation from naming to definition versus reverse speech, so greater for this condition than this, and for picture naming versus scrambled images. And as you can see that there are only a few areas of the brain that consistently survive. And interestingly, the ventral visual stream survives. So here they are naming to definition. They're not seeing anything. There's no visual stimulus on the screen, just a crosshair. But yet, a part of the brain that is very intrinsically involved in image characterization, object discrimination, is activated. And when you do a conjunction analysis linking auditory and visual pathways, Broca's area, a very small part of interparietal sulcus, a tiny part of the inferior temporal sulcus, pre-SMA, and this ventral visual stream in ventral temporal cortex are what are active. And so we characterize this as the lexicosemantic network, that these areas converse with each other to convert this input into meaning. This is just the time traces from those areas like I showed you earlier. We can go back and show when these given areas are active, and then the imposition of this color scheme onto the brain. We also validated this by stimulation. So all of these patients, many of these patients underwent cortical stimulation either with implanted electrodes, with grids, or with SEEG, and we identified which areas in the brain were active or were disrupted during specific tasks. And again, you see the same areas survive with stimulation mapping, different way of assessing the same function as with ECOG. So one of my collaborators in this project was is a neuroscientist and linguist at the University of California in Irvine, Greg Hickok, and he has this model of a hierarchical state feedback control model. So from the same data, we asked the question, could we actually even look at interactions between motor and auditory cortex during speech production? So this is the same stimulus, a place with sand along a shore. The patient is listening to this, coming up with the answer. But while they're listening to this, you can extract the acoustic envelope. You can also extract the edges of this acoustic envelope, which is like a derivative of the envelope itself. And then you can relate that to the gamma power in the recorded ECOG, which is shown here in C. So this is the peak correlation between the acoustic envelope, and the high frequency signal. And it shows that it really, really sharply lines up in Heschel's gyrus, which is what you would expect. Heschel's gyrus is most important for you to understand what is being said. But the intertrial coherence in low frequencies, in this case in frequencies between 2 and 15 hertz, reveal a involvement of all of early auditory cortex, and in fact a preponderance to areas not necessarily in Heschel's gyrus. But importantly, and this is the salient finding from this work, when people are listening, these electrodes in transverse temporal gyrus are active, but the areas in planum temporale behind it, when the individual speaks, shut down. So you see a sort of a dichotomous behavior between posterior and anterior electrodes in the superior temporal plane during listening and speaking. And this points to some very fundamental mechanisms of feedback to the planum temporale, presumably from speech motor areas during speech production, where these areas are no longer active when you're actually speaking. And it gives us some insight into how motor cortex suppresses your own auditory cortex or your own auditory processing while you're speaking, except for the fact that you still want to detect errors. And perhaps only when errors are detected this area comes online, and when it's not, when there are no unexpected articulations, it is suppressed. I said earlier that the most important value of ECOG is not just to create these topological maps of where things are happening, but to understand how they're happening and how brain areas interact with each other. One way of studying this is to look at states of the brain. So between me looking at a picture of say a balalaika and coming up with the word balalaika, many things must happen in my mind. I must break down that image, I must come up with potential answers, I must select, and I must then articulate. And while I'm articulating, I must then check to make sure that what I'm articulating is indeed correct. For me to do this, my brain has gone through multiple states. So the question before us is, can we decode these states? Can we look at interactions between the brain areas for decoding these states? And we worked very closely with a computational neuroscientist at Baylor and at Rice named Zach Pitkow for this work, creating an autoregressive hidden Markov model to study the interaction between brain areas during these tasks. And here is an example of these states. Here is time 0. This is now no longer gamma power. This is purely a representation of interactions between these 6 electrodes, 4 on the lateral surface and 2 on the ventral surface. This is a multivariate autoregressive model, okay, which looks at interactions between brain areas and then summarizes those as states that fluctuate from one to the other. And so very, very clearly you can see that the brain goes through these, at least these 6 electrodes go through these 3 states in the process of naming something. We're working on integrating all of our data sets of 100 plus patients into this so that we can come up with a comprehensive and complete representation of these states. And then more importantly than that is, how do these states fail? How do we get to the point of having a tip of the tongue phenomenon or not being able to come up with the right answer? Where does the state get degraded? What areas are responsible? And how can we potentially then come up with remediation? This is a representation of each of these 3 states. I showed you these states in red, yellow, and green. So this is the red state. It's the visual processing state. Yellow is the language processing, and green is articulation. And this was done over 7 patients. It's very consistent in these 7 patients. And as I said, we're now integrating this with a much larger cohort. We hope ultimately to take this kind of classical model of Indefry and Levelt, which is driven by psychometric data, and relate this then to these neural time series data. Moving from picture naming and listening to stimuli to reading. Reading, I think, is an even more impressive human accomplishment than speaking. Speaking gets my thoughts from my mind to yours. It's a way of almost of telepathy of my brain to yours. But reading is a way of doing telepathy across time and across space, where I can write something and send it across to you, and you can pick it up at your own time and read it. And again, my thoughts are transmitted to you. And we don't give these basic functions the importance, I think, that they deserve. But really, 10,000 years is as old as reading probably is. And it's an incredible accomplishment for a primate such as us to decode and to represent symbols. So all of these words are apple. You can all read them as apple. And you don't need any special training beyond what you did in kindergarten or maybe elementary school to recognize that those are all the way that apple could be written. Our models for reading are also derived from lesions and functional imaging. They also look very much like arrows and boxes. And so this is actually Stan DeHaan's model. He's one of my collaborators on a project now, and more about him soon. And this gives us a starting point to see, well, what are the kinds of systems involved in reading and how should we study them? So we first did a very simple task, a word stem completion task. We asked people to generate a verb from an incomplete word like this. So this might be burn, this might be catch, this might be bite. And the patient is looking at the screen, a single word is flashing, and they have to come up with the right answer. Similar to before, this is the heat map of where the electrodes are. These are the areas that we then select for our sub-analysis. This is part of the reading area in the brain, visual word form area. This is motor cortex. This is motor cortex plus Broca's area plus sensory cortex. And looking at all of these electrodes from these areas that are active, we can then create these time traces like I showed you all earlier. This is time locked to the onset of the stimulus. This is time locked to 0 is articulation. As you can see, the areas in motor cortex are active at the end, here in purple and in orange and in turquoise, whereas the areas in vision are involved early. So I showed you, and I probably should have shown you this slide earlier. This is Clive Granger, Nobel laureate for economics in 2003. His work underpinned much work at Wall Street, led to all of these quantitative measures of estimating when the next crash would occur. It failed miserably because of unpredictable events that we now characterize famously as black swan events that prevent these kind of models from being extended indefinitely. But in neurosciences, there is perhaps significant value to the work of people like Clive Granger, where a time series X is said to be Granger causal if it can be shown through a series of tests on lag values of X, along with lag values of another time series Y, that X provides significant information about the future values of Y. So then X therefore causes Y, right? I know it's a very complex explanation, but it's simply that one electrode at one point predicts what should happen at another electrode at a subsequent time point, simply put. And extending that to intracranial data and to these data, we showed that really all reading at this type of reading of completion of a word where there's a lot of interaction of thinking of what the word should be is really extremely top down. You're not really reading from your visual areas up, that it's your brain that imposes, your prefrontal cortex that estimates what is likely to be occurring from very early on. This is right very early, even before you would imagine that the individual has had time to fully read the word. The process is very top down, and we found this now with various other analyses. Similar to the data I showed you earlier, we can break these down into network states. Here are 4 separate states that originate during this early reading process, time locked to word presentation. We find these processes to be similar to these metastable attractors that others have hypothesized should occur in the brain, that cognitive processes should get to these metastable states, transiently stable states in which a computational goal is accomplished, and then the brain moves on to the next state. We furthered this reading work by, with the help of the NIH, with this UO1 mechanism that we are now in its 3rd year, that studies language not just as a separate system, one experiment versus another, but a conglomeration of experiments, very basic to very complex, in the same individuals, try to collect as much data as possible over a span of several days to then integrate the most basic with the most sophisticated language processes, from the earliest visual, phonological, to the syntactic, semantic, and articulatory processing stages. I'll show you a little bit of data from that if I have a few, maybe 5 minutes. Here are individuals looking at a variety of different stimuli. These are false fonts, infrequent letters, frequent bigrams, quadrigrams. This is just a localizer task, if you will. And then they're also looking at this. This is Lewis Carroll, another fantastic human being, right? Had epilepsy. Some of you may or may not know that. Had occipital epilepsy. Things got big. Things got small. So he created Drink Me, Eat Me. Created a little girl falling down a rabbit hole, getting trapped in a place with big people, with very little people, right? But he also created Jabberwocky. Jabberwocky is a poem that he wrote. The Jabberwock was the name of a dragon. And Lewis Carroll was actually Charles Ludwig Dodson. He's a mathematician at Oxford, clearly a sophisticated human being to do all of this in one lifetime. But Jabberwocky is a communication of syntax, which means grammatical sense, without meaning. So meaningless sentences that somehow seem to still be a sentence. So here's an example of Jabberwocky. This prages twig is truer than this portus. You know I'm saying something about this relative to that, but you don't know what I'm saying. But it's still, interestingly, the brain still tries to understand this, and Jabberwocky is helpful in helping us understand how we parse language, how we break down sentences into individual components. Now, the way we present these stimuli is by single words flashing on the screen, because we want to control exactly when a given word is presented. So we call this RSVP, or rapid serial visual presentation. It's not words strung along parallel to each other on alongside each other on the screen. And then we do time-locked analysis, in this case to words 3 to word 8. And here we see that the Jabberwocky words are much more active than the real words in this visual region anteriorly, as well as in the more canonical visual word form area that's slightly more posterior, where we have less electrodes at this time point. This is also true for content words, so anything that requires much more engagement by the brain seems to be much more active. This is a bit different when you look at these very simple words. The localizer task actually shows sort of a different result, that there is greater activation based on number of letters for, say, Jabberwocky content words. Here for sentence content words, this is tracking the number of letters in each sentence, but I'm going to jump through this. But right here you can see that the most significant component here is actually the word frequency effect, not the bigram frequency or the word class. I'll leave you with another thought. I showed you syntactically valid sentences without semantics. Here's another type of class of word like that. This is a famous sentence that actually was presented as a contest to people. Someone had to make sense of this sentence. So I'll let you dwell on it for a second. Colorless green ideas sleep furiously. This is Noam Chomsky, famous for a variety of things in psychology and language. But you could actually make sense of this. It can only be the thought of verdure to come which prompts us in autumn to buy these dormant white lumps of vegetable matter covered by brown papery skin, and lovingly to plant them and care for them. It is a marvel to me that under this cover they are laboring unseen at such a rate within to give us the sudden awesome beauty of spring flowering bulbs. While winter reigns, the earth reposes, but these colorless green ideas sleep furiously. So yes, it is still winter in DC. Spring will come soon. So I'd like to thank all of the people who make this work possible. I know it's been a bit of a barrage of information the last few minutes. There's the postdocs and graduate students and others in my lab, and collaborators, Nathan Crone at Hopkins, Stan Dehaan in Paris, Greg Hickok, whose model you saw also along with Stan's, Zach, who's done all of our analysis for state derivations, Behnam Ajang, who's electrical engineering chair at Rice, former chair, And then this is our epilepsy program, without whom none of this would happen. None of these patients would ever get seen or managed. And very recently, our institution has invested heavily in creating the Texas Institute for Restorative Neurotechnologies. We have several goals in the education and the promotion of the care of epilepsy, but also in the development of a variety of new databases and neurotechnologies. We hope that this will be transformative. So I'm supposed to put on some slides about CME credit, and I guess I have a time for a few questions.
I had a question about when you put the coverage map of the aggregate patients, 106 patients. For the SEEG, do you only consider the contacts inside the cortex, close the cortex, or there's a diameter around the SEEG contact?
Yeah, it's a very good point, and I think increasingly, we recognize that SEEG electrodes will also, you have to be very careful about where you assign the recording zone to, because they can record from the other side of the sulcus sometimes, from where they are. It's a much more attenuated signal, but it can occur. So what we do is we create a diameter of 5 millimeters around the center of each electrode, and then we constrain it by the cortical ribbon. So this is where precise localization becomes important, and then restricting it to that cortical ribbon becomes important. So and that spread can be greater along the cortical ribbon than it is along the white matter.
And for the grid, you consider just the surface, right? The fold, your area of recording doesn't.
So I would point you to those papers, especially the paper by Karipashoglu in PLOS One and another one in Neuroimage, that talk about how to assign recording zones by topology rather than what we call a geodesic spread rather than a Euclidean spread. So it's not equidistant, but it's related to the actual folding characteristic of the surface.
Another question I had, and maybe you alluded to that when we were talking about the states. So there were still the same work you've done before, presenting a stimulus and then recording states to work like in relation to that stimulus or that action. Can you find the same kind of pattern in resting state as well? So it's like resting state sitting.
Yeah, so the resting state is always a state. I didn't point that out, but there's a fourth state actually, which is the resting state that occurs right after you finish monitoring your articulation. So in fact, that resting state occupies the largest proportion of the entire epoch. And what is very nice, I think, is that this derivation brings us back to exactly that same state. Just based on network connectivity, and I suspect it's because the connectivity really degrades to almost nothing soon after you finish speaking.
Thank you very much. Another question?